Hi to everyone that's joined so far. We're just waiting for the attendees to filter into the session before we begin. Okay, it looks like everybody's here. So hi everyone, welcome to this EMQN guest webinar. We've got some really great speakers lined up for you today, and we hope that their sessions will be really interesting and informative for you. My name is Rachel Taylor. I'm your host for this afternoon. And so first, I just quickly like to run through how the session will proceed this afternoon. So in the next few moments, I'll give an introduction of the technical aspects of this webinar. There will be parts of the session where you can interact with us, and I'm just going to give some instructions about how to do that. Then our operations director, Nikki Wolstenholme, will give a full introduction of our guest speakers. Following that, our first speaker is Dr. Peter Causey-Freeman, who'll give his seminar, which will be followed by a short Q&A session. Then after the Q&A session, Professor Johan Den Dunnen will host a HGVS quiz, and following that, we'll give his seminar with a short Q&A session at the end, and then Nikki will close the session at around 2 p.m. this afternoon. So just to remind everybody, this webinar is being recorded, and we will be disseminating a link to the recording to all registrants following close of session today. And we ask that you refrain from using any patient identifiable information during the discussions. I also have to mention that the views expressed in this webinar are those of the authors only and do not necessarily reflect the views of EMQN. So there are elements of the session where you will have the opportunity to ask questions, and you can do that in one of two ways. You can submit a written question using the Q&A button in the toolbar that you should see on your screen. Or if you'd prefer to ask verbally, you can use the raise hand function, and when an appropriate moment arises, I'll ask you to speak. There will be a quiz following the Q&A session after Peter's talk. For this, I'll launch the question and you should receive a pop-up on screen. If you could select the answer that you think is correct and then press submit, that would be really helpful. I'll be able to see the number of participants, and I'll display on screen a survey of the results at the end of each question. I'd just like to mention as well that at this point that all answers are anonymous, so you don't need to worry about getting the answer wrong. We hope that this is more for educational purposes. And so with that, I'd like to ask Nikki to introduce the speakers.
Hello.
You're muted, go on, it's okay.
That's lovely. Hopefully I'm unmuted and you can see my camera now. Right. Good morning, everyone, and thank you for all attending this webinar. It's really good to see so many of you here today. It's been several years since we were able to have a guest speaker at one of our assessment meetings, and we thought this year might be a great time to bring back that tradition, even if it's in a virtual format. I'm sure that the programme we've lined up for you today has generated some curiosity. In fact, we thought it was so interesting that we invited all of our assessment teams, not just the ones who are about to assess the inherited EQA schemes. And we've got some real hot topics on our menu today. Firstly, I'm very pleased to introduce Peter Causey-Freeman, who's a lecturer and researcher in healthcare sciences, specialising in clinical bioinformatics at the University of Manchester. Pete also teaches on the scientist training program for the NHS clinical scientists in clinical genomics and clinical bioinformatics. So he's helping to train the clinical scientists and bioinformaticians of the future. However, you might know Pete better for his work in developing the genomic variant nomenclature validation software, Variant Validator. Today, Pete is going to talk to us about reference sequences, which I know will be of great interest, as there's been a lot of discussion on forums about the benefits and some worries about the introduction of a new set of reference sequences, the main select transcripts. Listening in on these discussions, particularly regarding the BRAF gene, it's clear that now is the right time for the engagement with the diagnostic community to better explain the concept of main, why there are problems with LRG, and for the community to start to have a debate about recommended reference sequences. For our second speaker is probably familiar to all of us, as I'm sure you will have seen his name on the HGVS website or sent him emails querying variant nomenclature guidance. Or is that just me? Anyway, I'm excited to introduce Johan Den Dunnen, who's a professor of medical genomics in the Department of Human Genetics and Clinical Genetics at Leiden University Medical Center, whose research is focused on the use of new high-throughput genome technology for both research and diagnosis of genetic disease. It would be fair to say Johan is the driving force behind the development of HGVS nomenclature. He established and continues to coordinate the LOVD platform, and he's a committed board member of Hugo. For most of you, I know that naming of genetic variants is just an integral part of your day-to-day work. But for some of us old timers who've been around since before the CF gene was discovered, we remember the problems encountered with tracking familial mutations in the days when nomenclature just wasn't standardized and variants were reported without listing the transcript used for numbering, and the cDNA did not start from the A of the ATG. So it was quite interesting when you did a predictive test for a rare disease, and it could involve quite a lot of emails to be sure you were even testing the correct exon. Nowadays, we think we're experts in HGVS. Well, that's until we try and mark someone else's attempts at it, or we need to deal with an appeal. Then we start to wonder if maybe we found an exception or if the guidance may have changed. So HGVS isn't always simple, and at EMQM we do try and take a pragmatic approach to our marking. However, I still like to think of us here at EMQN as the guardians of systematic nomenclature for the clinical diagnostic community. We know it's not always easy, and we do have our difficulties in marking it, particularly around describing uncertainty or when there's multiple changes. And we need to do it in a way that's concise, consistent, and meaningful. So today, Johan's going to host an HGVS quiz, which we hope will be quite a bit of fun. He's also going to provide us a bit of guidance, and both the speakers will deal with some of your questions on the correct use of HGVS and transcript use. So Pete's going to kick off today to explain why the diagnostic community might want to adopt MAIN in the genomics era, with reference to some of the benefit and some real world problems with the existing reference sequences, and in particular our favourite LRGs. So I'm going to pass you over to Pete now.
Okay, I think my video should be on now. Is that working?
Yeah, we can see and hear you, Pete. That's great.
Thanks. Just quickly, I'm going to have to ask, I'm having some unfortunate Zoom issues at the moment, so Rachel's going to steer my presentation for me. So Rachel, if you don't mind sharing your screen, put that on for me, please.
I am sharing. Oh, sorry, 1 second. Here we go. Should be able to see it now.
I'm still seeing the overview slide, the first slide.
There we go.
Yeah. So just to get started, I do have more than one shirt. It seems I'm wearing the one that I actually had in my profile picture was embarrassing. So next time I'm going to plan ahead a little better. But thanks very much for the introduction, Nikki. Could you just go to the next slide, please, Rachel? So I'm not going to, you've already been told about me. I'm not going to dwell on that for much longer because we've, quite a tight schedule, but I do maintain a research profile page. The slides I'm sure are going to be shared. So if anyone wants to know more about me or contact me, please feel free to do so. My details on here should be correct. Next slide, please. So the idea of this little talk I want to give you is that I really want to try and start taking the fear out of this new and fangled thing that we're calling the main select transcripts, the main plus clinical transcripts, because I've been bouncing this with Nikki for a while now, and it seems that the more I look into this, the less scary it's getting. And putting this talk together has actually been very helpful for me to understand what the background of main is and why it is that we do need to start making changes in the actual way that we report in terms of the transcript and reference sequence selection. So these are the topics we're going to cover. You'll see this go through. I'm going to illustrate some of these with some scenarios, which are clinical examples of where poor transcript selection has caused problems in diagnostics. And there's going to be a few take-home points, which is why LRGs have become problematic and what the next steps are. So the next slide, please. And so this is the first topic. So basically, the first thing I want to cover is what exactly is a transcript reference sequence or any reference sequence. I'm going to focus this particular slide on a reference sequence taken from RefSeq, the NIH database. And effectively, a reference sequence is really quite simple. It is an identifier, a standard identifier that never changes. At the bottom of the page, if you go to one of the records, you will see a sequence, a genetic sequence. That never changes. But then you've got all this stuff called metadata, all this additional information that tells you about that sequence. So it'll tell you things like, where the exons are, where the introns are, and that kind of thing. So you've got a lot of additional information there, publications and evidence. So next slide, please. Well, the take-home point is that a reference sequence is effectively two things that really matter. It is an identifier and it is a sequence. And the way in which this then becomes a gene when we actually look at it on a genome browser is that reference sequence is aligned using a bioinformatics algorithm to a reference, so a genomics or a gene reference sequence. So this is one thing to be careful of, is that just because a gene, so a transcript may have one structure when aligned to a genome build, say GRCH37, or maybe aligned to GRCH38, or maybe aligned to a gene, the actual structures of that transcript can change. So it's very important to know what the transcript sequence has been aligned to. And I'll talk about tools that we've got for that. So just go back to the last slide one more second. Just one point I'm going to make is, and also you can have, there have historically been problems. This is now something that's gone away, thankfully, that in the past, depending on which company or which which organization was displaying the data, you sometimes had differences in how the transcript was aligned even to the same genome build. So there has been a lot of problems with this in the past. So we are very much pushing towards some kind of standardization here. It's a big standardization, harmonization is a big field, a big topic in the genomics field at the moment. Next slide, please. So I've said that the idea is that you take a transcript reference sequence, you align it to a genome. So there are two ever so slightly different approaches, the one that RefSeq use and the one that Ensemble use. And I'm going to just quickly focus on these with a little bit of an overview. So the idea for the RefSeq transcript is they have got an idea of what the transcript sequence is, and they go along and then align that to the genome using their software. This means there can be sometimes some mismatches in the alignment. These may be sequence mismatches or slight length mismatches within the exons. There are some very, very rare cases in which there's been some problems with their reannotation, and this is why the HGVS in its new, the sequence variant group in its new reference sequence guidelines is starting to push everyone to start at least reporting a genomic variant description, so that if any of these issues occur, we get back to the anchor point and we can figure out what's changed over time. Next slide, please. The Ensembl approach is ever so slightly different. Ensembl really, technically it's a reference sequence archive, but more it's a genome annotation archive. So the difference with Ensembl transcripts is that they are based on genome annotations, therefore their sequences will always match what's in the genome. There'll be no sequence discrepancies. Next slide, please. So let's talk about LRGs. Well, LRGs, believe it or not, are very much based on RefSeq reference sequences. Ensembl as well, but it's much easier to describe in the context of RefSeq, and I've got some slides later on that will show you how this works. But in this example, you can see we've got transcript LRG1T1, and that transcript is pretty much identical to the RefSeq transcript NM_88.3. So those can almost be used interchangeably. And again, I'll describe how Main has made this easier for you and describe some techniques that we've got to actually get you to the right place. Some of these LRG transcripts, the RefSeq transcript equivalents, have been mapped to both GRCH 37 and 38. Now, this is ideal for you if you want to find that genomic description. Next slide, please. But unfortunately, some of the LRGs are really quite old. In this particular instance, LRG16, the reference sequence, equivalent reference sequence from RefSeq is NM_0s22.2. That has never been aligned to GRCH 38. It's only aligned to GRCH 37. Next slide, please. And the thing is about this is that LRGs were designed to be static reference sequence. You know, we want to keep them simple. We don't want them changing that much. So updating the sequences and this kind of thing means that if you want to update the sequence of the transcripts or anything like that, you have to change the sequence of the actual LRG itself. You have to create a brand new record. Now, theoretically, RefSeq could align the 22.2 the LRG transcript to GRCH 38, but it's kind of a big ask for them because they've already, this had got replaced before GRCH 38 was made by the 0.3 version. The latest version is 22.4, so it's unlikely they'll ever release those alignments. There's another slight problem with LRGs in that, unlike genomes, they don't have annotations for every single clinically relevant transcript to them. So the problem is that if you want to map between one LRG transcript and the other, you cannot go directly to the LRG reference sequence and back again. You have to go to a genome build and then back onto the transcript. So there is actually some problems with what the content of LRGs and, you know, what are the clinically relevant transcripts, and these do need to be considered, which I'll talk about shortly. Next point, please. So something about sequence identifiers. Now, this follows on from a point that Nikki was making back when you weren't describing, you know, telling everyone what sequence would be used and this kind of thing, and the problems that kind of occurred. So the next slide, please. So one of the things the HGVS nomenclature stresses is that you should use the complete and unique reference sequence identifier. Ideally, provide it with your description, but at least enclose it in the document that you're writing. This is essential. Now, these are some examples I wanted to give. I was going to ask the question, which of these statements is correct? Chromosome M for GRCH 37 is the same as chromosome M for HG19. Now, I know for a fact that one's not correct because those are actually different and very few people actually know that. In fact, UCSC, when they created HG19, weren't aware because RefSeq didn't tell them. Chromosome 1 GRCH 37, the same as chromosome 1 GRCH 38. Again, I know that's wrong. But these others, I couldn't remember the answer. And the simple fact is that because I haven't been given a unique reference sequence identifier, it would take me ages to actually go back and check this. And that's the point. You have to provide the sequence identifier, the unique identifier for the sequence you are working with. Otherwise, you can face problems. Next slide, please. And this is quite a common one. We see examples of this all the time. Now, Element Visual, I'm not knocking it, absolutely fantastic software. Many of you may have used it. Many of you will be in contact with labs that do use it. But the problem is Element Visual doesn't give accurate reference sequence identifiers for genomic descriptions. So you can see here we've got chromosome 6, GRCH37. I've seen this so many times, being shortened to chromosome 6, lose the GRCH37, and as soon as we get to that point, we cannot infer the reference sequence. That is a description that cannot be used. So what we should be doing is using the unique reference sequence identifier, and that is the reference sequence identifier for the GRCH37 version of chromosome 6. It's different. It's got a different identifier in GRCH38. Take care of your software as well. Alamut doesn't always get the nomenclature right, especially for genomic descriptions. So using a tool like Variant Validator to check that your description is accurate is also advised as well. Alamut's great, but it's always worth using another tool to double check. Next slide, please. So just a little thing about stable and permanent. Now, some of you may have seen these kind of descriptions of transcripts. So you've got Homo sapiens, BRCA1 DNA repair associated, BRCA1 transcript, a long description of the actual transcript itself. Now, this description is mutable, and by mutable, I'm going to use that term. It means it can/will change over time. The stable identifier is the NM_007298.3. This stable identifier will never change, and it only ever represents a single sequence, which will also never change. So these two are intrinsically interconnected, and in order for one to change, the other needs to change. So say, for instance, we decided, and you'll see this a lot in LRG annotations, is that this transcript is identical to this RefSeq transcript, but with the poly A tail removed. Well, what if we actually wanted to create that transcript without the poly A tail? Well, that means we've got to change the sequence. If we change the sequence, we must change the reference sequence identifier. Next slide, please. Same happens with genes. We have mutable aspects and we have non-mutable aspects. This symbol here, now a bit of nomenclature here for you as well. I keep seeing gene symbols referred to as gene names. No, they are not gene names. LEPR1 is a gene symbol. A gene name is leucine proline-rich protoglycan 1. The actual stable IDs for this are the HGNC numbers there, and these can be found on the website genenames.org for those that don't know. Both of these genes have now been updated. These symbols are now out of date. The top one, the new symbol is P3H1, and this was changed for reasons that it was unseemly to call someone with short stature because of a genetic disease, leprechaun. There was a unfortunate connotation of that, so they changed the name of the gene for politically correctness reasons. March 3 was changed because a lot of clinicians love to use Excel, and if you type March 3 into a cell in Excel and press enter, it becomes March the 3rd, and it really confuses the software, and then we lose the gene symbol entirely. So this one has also been updated. But even though they've been updated, the HGNC identifier has never moved. Next slide, please. So I'm going to give you a little bit of a clinical scenario and tell you a little bit about where some of this transcript selection has gone wrong. So this was actually a case that I was involved with a colleague at the University Hospital of Leicester quite some time ago. There was two sisters. Elder sister had advanced breast cancer that they suspected to be familial. The younger sister was seeking options for preventative treatments. They also need to consider what might have been the potential impacts on her children, etc., if they've inherited a potentially pathogenic variant. A variant had been identified in the BRCA1 gene. This is the genomic description, and it was at the time classified as uncertain significance. Next slide, please. Now, the bioinformatics predictions are made on a transcript by transcript basis. In fact, all clinical decisions for each transcript are made on a transcript by transcript basis. In terms of the informatics, there was two really strong candidates for wondering whether this particular variant was going to be pathogenic or not. We've got the so-called canonical reference sequence, which is 72994.4. This is actually annotated in the LRG. This is LRG T1, so it's the transcript 1 version. Then we've got this alternative one, which is transcript variant 4. Next slide, please. So the transcript variant 4 actually does have strong bioinformatics evidence that suggests that in the context of this transcript, there is a good chance that this variant could be pathogenic. The ACMG guidelines do say that laboratories should consider the impact on all clinically relevant transcript. Now, at the time, and this was several years ago, contacted the lab and they said, well, we don't actually predict on anything apart from the, for BRCA1, the LRG transcript, because, you know, that is the, you know, it covers all the bases. It's got all the exons and this kind of thing. So that's the one that we look at. So they don't use this, and they actually gave some kind of name like BRCA Delta 11B. That was what they said at the time, because it is a predicted transcript. And I'm like, well, I was really confused because they told me a name and I couldn't find that name because names, as I said, are mutable. And I didn't think there were that many predicted transcripts for this gene. So how did I go about tracking it down? The next step, please. Next slide, please. So what I actually did was do a RefSeq search, and when I searched the RefSeq database, a top hit came up, and it told me that this was the transcript variant 4 that I'd mentioned earlier, this alternative transcript. And you can look through the revision history for this transcript. You can see that this has had previous names in the past. Although the identifier is the same, and there may be versions of that identifier, it has had different names. And this was leading to the confusion. So this is why we need to know the identifier. Which transcript is it we're actually talking about? What sequence do we actually talking about? And as it turns out, this was not a predicted transcript. This is actually one that RefSeq considers to be a very good quality transcript and clinically relevant and biologically relevant. Next slide, please. So the thing is that we need to start getting this kind of thing right, because, you know, we are essentially dealing with lives. A variant of uncertain certificate prediction interpretation meant the patient couldn't, no, so the patient's sister couldn't be given preventive treatment. Her sister was critically ill at this point, and unfortunately since then has passed away. The clinical geneticist was under a lot of stress because of the extreme marginality of the case, as you can imagine. So next slide, please. So the bioinformatics evidence, you know, on its own is not particularly strong. We know that it doesn't score very highly, but the clinician managed to do a lot more work thinking about this other transcript, thinking about this variant, and got a lot more biological evidence. And the variant was eventually raised to being pathogenic, reclassified from a hot 3 to a level 5. And just to let you know how marginal it was, the bioinformatics evidence on its own, even though it doesn't have much weight, was enough to reclassify this variant from a hot 3 to a 4, likely pathogenic. And that obviously there's different options available for each level of the diagnostic spectrum we go up. So the simple fact is that transcript variant 4, is something that is proposed to be clinically relevant. And the difference is that interpretation in the context of that transcript compared to what we'd class as the canonical could make a difference. And so next slide, please. And this is a little bit of an issue with the LRG at the moment. As I said previously, the LRG, I believe the LRG 292, the BRCA1 LRG, has only still got one reference sequence transcript in it. And the idea for LRGs was to be, you know, what we actually wanted at the time was a reference sequence that had annotations of all the clinically relevant transcripts that we ought to be considering, because selecting which transcripts you actually want to consider in the clinic is very difficult. There's no real consensus between labs. LRGs really should have been, you know, should have sorted this out for us, but unfortunately they've fallen well short of this. And the problem is now that a lot of them are really old and there's been more evidence provided, but the LRGs predate these. For example, at the bottom of this slide, you can see expression profiles of different transcripts in the BRCA gene. And actually the second from the bottom on the right-hand side, you can see that the transcript, that's the kind of expression you'd be expecting from the full-length transcript. That's the one that's equivalent to the LRG1T1, sorry, the LRG 292T1 transcript. The expression is really quite low. But actually, the transcripts that are lacking exons 9, 10, and 11 are very much more expressed in breast cancer. So there is evidence to suggest these are highly expressed and clinically relevant. So we do need to start having more clinical data and actual biological data put into the decision making for transcripts. Next slide, please. So what is main and main plus clinical, and why should we care? Next slide, please. Well, it may seem scary because this is suddenly some new thing that's coming out, and the idea of it is a matched annotation between NIH and Ensemble. And this seems quite scary. You've got these two big bodies that make transcript referencing, well, that make reference sequences getting together and trying to make something that's clinically relevant for your use. Just a couple of points I'll take home. I've highlighted this, you get them in the slides, but the main points are that is the idea is that the main select, the idea of main select is to have a canonical or reporting transcript for every single gene in the human genome. And then you've got this thing called main plus clinical, which aims to add all the clinically relevant transcripts. Sound familiar? That's what we wanted from LRG. That was the idea of LRG. Next step, please. Well, to be honest with you, having looked at this in lots of detail, don't be afraid of main. It is effectively LRG, but updated for use in a genomics era rather than in the era where we were still aligning sequences to genes. LRG was an Ensemble NIH project, just like main is. Whereas LRGs had poor coverage, only 15% of genes had an LRG, main has already covered 90% of genes with at least the canonical, the select transcript. LRGs are static. They were designed to be static because they were designed against gene reference sequences. So they didn't allow evolution of the genome. Well, the simple fact is the genome, the latest build of the genome build, latest genome build is really pretty good. So the argument for having separate genes these days is very much, it's no longer needed. And the fact is we align now in the genomics area, we align our NGS against genomes and not genes. But the nice thing about the main transcripts is they've considered the fact that the genome may change slightly if there's another build. So if we do bring out a genome, GRCH39, there may be a slight change. But because they've taken this into mind, the reference sequence, the actual main transcripts shouldn't change. They shouldn't have to be revised because they've actually got the for the genes where there's issues, they actually build these on an alternative genome assembly which can be merged, and that's how they make the new genome build. So they are actually in line with what would be coming next. So they are built to be future-proof, unlike LRGs, which were designed during the days of gene sequencing rather than genome sequencing. Next slide, please. And also, another reason I'm saying don't be too afraid of these is that part of the process to make main transcripts is for the reference sequence body, so in this case RefSeq, to identify what it thinks to be the select transcript, the main transcript or the main select transcript in its own right. So the LR, in this case, is called the RefSeq select. The bit I've highlighted here shows you that if there's actually been an LRG transcript made, reference standard made, they will consider this. They will just carry this through to their select project and it'll automatically incorporate into main. The nice thing about this is that what they'll do is they'll take that transcript, and whereas in the LRG record you say this is the same as this transcript but with the poly A tail removed, they're just putting the latest versions in where they've actually done that. They've removed the poly A tail and they've done things like they've polished up the sequence. So there's not going to be much difference at all between the LRG sequences you're used to and what's going in the main project. So it's a lot less scary than it actually sounds. Next slide, please. So, as I've said, main could definitely be thought of an evolution of LRGs. LRGs, as I've kind of alluded to this previously, they've never really been independent reference sequences. They've actually been reference sequences designed by generally RefSeq and sucked into a third-party database and annotated. So for this transcript, for this gene, the COL5A1, we are in a situation where the latest version of the RefSeq transcript would have been a better version of LRG 737T1 because it does have the poly A tail removed. So it would have actually been nicer to have had that in the LRG, but unfortunately we can't do that. The same thing when it comes to this particular gene's got 2 transcripts, and again, the 737T2, the upcoming version, the version of the next reference sequence for this gene that comes out, which is an alternative transcript, would have made a better T2. It would have been really nice to have that integrated into the LRG, but unfortunately we can't have that because of the way that LRGs were designed. So the nice thing that main's done is it means that you can take a variant description and simply remove a RefSeq identifier and put the equivalent ensemble identifier in place. Why is this important? Well, a lot of you will be seeing data coming from Genomics England, things like in the UK, around Europe. The use of ensemble data is becoming more and more prevalent. It would be nice for us just to have a lookup table and say, well, actually, for that particular reference sequence, we could put this RefSeq one in. It's exactly the same, and we don't have to worry about it. You know, the annotation is exactly the same transcript, just with a different name. Not a problem. So that's why main will really help us. Certainly if you work with Genomics England data, that will really help you in the future. And unlike LRG, the idea behind main, well, this was the suggestion anyway, is it did intend to include a comprehensive set of clinical transcripts, not just a couple of transcripts or one transcript. So it really wants to nail this. What are the clinically relevant transcripts? We do need this in the community. Trying to teach this, trying to tell NHS students how to select transcripts based on current evidence is really difficult. And that's why we've got no consensus between the labs. And this is why we have these problems of variable reporting. The thing is, LRG hasn't tackled this. It's maybe made it better for a couple of genes, but not many. Main is trying to push that ethos forward and make this better for all genes. Next slide, please. So some of you might be wondering, well, you know, why do we need main plus or main select, which is the canonical transcript, and main plus clinical, which are these alternative transcripts? So another quick case study. This is a really interesting case that I came across a while ago. My colleague Raymond Dalgleish at the University of Leicester does a lot of work online around Ehlers-Danlos syndrome. Next slide, please. One of the weird things about the Ehlers-Danlos syndrome is that we have the gene COL5A1, which is very much involved in this, but most of the COL5A1 variants that you find typically give a dominant effect, so due to haploinsufficiency. But in this particular instance, in this gene, we had a variant in a variant that was effectively being transmitted in a recessive form. So you're kind of like seeing an inheritance pattern, which is indicative of recessive inheritance of this classical Ehlers-Danlos syndrome in COL5A1 gene, which is really, really unusual. Next slide, please. Well, COL5A1 actually encodes two proteins, protein variant 1, protein variant 2. The isoform A, so the protein isoform A, has a total of around 70% of expression, and that retains an exon 64A, whereas isoform B actually has an independent exon. So it skips exon 64A and chooses a different exon in the gene. So those exons are not overlapping. And this is why we need a main plus clinical, because we need a representation of both transcripts, which one incorporates the isoform A exon and one incorporates the isoform B exon. And the simple fact is, next slide, please, that this particular variant, this genomic variant that was identified in this patient, proved once and for all that this transcript, this alternative protein, was clinically relevant. If you actually look at the variant in the context of what would be the select transcript or the canonical transcript, main select, LRG, whatever it is, T1, it's deeply intronic. However, in the context of the second transcript, which would be the main plus clinical, LRG T2, and in this instance, the LRG COL5A1 LRG does have this transcript. It's actually a frame shift, early termination, loss of function. You're losing about 30%, around 30% of the protein. So you get in this kind of recessive recessive issues. So say, for instance, you lost one copy, you lose about 15% of the protein. That's kind of going to be okay. You're not going to see Ehlers-Danlos syndrome, but this individual is inheriting twice. It's been heterozygous. They've shown the symptoms of the disease. So this is why we need to have a comprehensive set of transcripts for each gene. Next slide, please. I'll skip over that one. Another thing that Main does, and this is something that came up in the discussions I was having with Rachel and Nikki when I was putting this talk together, is that Main does remove reliance on third-party annotation. So as I said previously, LRG is great. The simple fact is that, you know, if you actually break it down to its component parts, it's effectively an additional database that takes third-party reference sequences, puts them somewhere and annotates them third-party. So let's just have a quick look at this example. This came in as a bit of fear that came from the genomics community, talking about the BRAF gene that Nikki mentioned earlier. And the fear was that standardizing on the main select transcript would result in clinically recognizable variants that are historically reported in a shorthand nomenclature being missed because the numbering in context of the protein and the transcript would change. Now, what was inferred here is that the reference sequence of this would be the LRG299T1, which is equivalent to 4333 point some, I think it's 0.4 actually. This is the 0.6 is the most up-to-date version, but that would give you a protein level variation of valine 600 glutamine. So V600E. This is the shorthand that's often used, V600E. And it was what we were worried about is that if we adopted main, all the reports that had used V600E would suddenly become unrecognizable to the community because you'd suddenly end up with the position V640E, which is a very subtle change and could very, very easily be overlooked and clinical decisions may be missed. Because you know that Nicky will be able to tell you the actual importance of the variant. I'm looking at the nomenclature of it, but that is something that is actually used as clinical decision making. And that was because the main transcript was going to be this. Well, that if you look at the LRG record, it tells you the main transcript is going to be this RefSeq identifier here at the bottom, the 1374258.1. Next slide, please. Yeah, that's not going to happen. It's actually an issue with the LRG record. As I said, LRGs are third-party annotations, and the annotation in the LRG record is absolutely incorrect. If you click the link that says this is the main transcript and actually open up the first-party annotation from the RefSeq record, you'll see that this is actually main plus clinical. It's not main select. It's not the canonical transcript. This is something that I'll tell you all now from my experience, is that if you ever look at cross-referencing data between reference sequence identifiers, I hate to say it, particularly if you're looking at ensemble data and it tells you this is the equivalent RefSeq, you are going to get bitten eventually because there are lots of mistakes in that. Always go to the source, never believe the cross-referencing data. And again, this is why we want you to start providing your genomic descriptions, because it's the only clean and effective way to map onto other transcripts. You can't just replace the reference sequence identifiers unless you know these are identical. And that's what main's doing. It's making the ensemble transcript and the RefSeq transcript identical, so you can quite simply slip off one identifier and put the other on. But in any other circumstances, that is not safe to do. Next slide, please. So, as Nikki mentioned, I'm developing this tool, Variant Validator, and we've put in some, I'm putting in some brand new features now, which will be released in the middle of this month, hopefully, which will help you update your current existing LRG-based variant descriptions to something that is the RefSeq equivalent of that description, and then how you can update to main. So I'll just go through that process now. Next slide, please. So those of you that haven't used Variant Validator, this is what it looks like when you actually. So there's a set of tools. If you say to validate a variant, you press the options available. If anyone has any issues and doesn't understand how to use it or anything like that, please do contact me. I gave you my contact information. I'm quite happy to walk you through this. I've actually got a video that I'm going to share with the group showing how I've done this process, but I didn't want to do live demonstration for the purpose of this talk because they always go wrong. I've been bitten too many times. So the idea is you put your variant description into variant validator. In this case, it's LRG 299T1 1799T to A. And then you would hit submit. Next slide, please. And the first thing that does is it shows you the HGVS equivalent variant descriptions. These I mentioned these earlier. So LRG 299T1 is effectively the same as NM 00433.4. So these are almost identical transcripts, but what we've done is we've done the validation from the LRG across and lifted it across to make sure it's still valid in the context of the transcript that the LRG was based on. So that is a quick way to update your LRG 299T1 into a refseq variant description format. The same happens with the actual LRG gene sequence itself. We've mapped it onto the refseq gene, and the same with the proteins. LRG 299P1 is the same as NP underscore 004324.2. Okay, hopefully that makes sense to everyone, but those are equivalent variant descriptions. Next slide. So how do you find the main descriptions, main select, main plus clinical? Well, the first thing you do is, if you go a bit further down the validation page, you'll see the genomic descriptions. What I did is I clicked on the GRCH38 description in the table, and that populated again this this input, these boxes for inputting sequence data, so sequence variants. And this is a brand new feature that's going to come online. This is not yet available. It will be made available very shortly, I promise, that I can actually now select specific transcripts for genes or genomes. I've provided, as I say, I'll provide a video that walks you through this process. So just to give you the additional information. But the idea here is that I've put in lowercase main into the select transcripts box, and then if I click submit, next slide, that would be. What it does is it only gives me back the transcripts that are associated with main. So what I've done is I've taken that initial description in the context of an LRG transcript. Variant validator's automatically mapped it to a refseq transcript that's equivalent. Then by going to the genome equivalent description, I've then been able to map that onto all of the transcripts available in our database, and then it's filtered them down from main and main plus clinical. It's nice and easy. If anyone wants to, like I say, the video will walk you through this. Any questions, please do contact me. We also have a contact us form on the Variant Validator website. Those of you that wanting something a bit more automated to update your databases, that kind of thing, if that's the way you want to go, we can provide resources. Yep, sending over a 5-minute countdown, that's fine, because I'm about to go on to my last slide, so that's good news. So one of the things I want to ask is that, you know, Variant Validator is very much a community-driven resource. One of the people who's really driven this forward has been Johan, because he keeps badgering me every time something goes wrong or he wants a new feature put in. And this is the kind of interaction we want with you guys. If there's something you think that might be useful, please let us know. If they think there's something broken, please let us know. Next slide, please. And this takes me to the final point. And why are we doing this? Why are we trying to harmonize? Why are we trying to get variant reporting right? And why do we need to start making this move now? Next slide, please. Well, as I've hopefully convinced you, and the biggest critic will certainly be Nikki, I'm hoping to convince Nikki that actually moving away from LRG to main isn't actually as big a jump as we originally thought. Kind of try to start thinking about main as not a replacement, but an extension of LRG, but just polished up and made fit for purpose in the genomics era. And this is important. We really do need to start finding a way of providing variant descriptions that are harmonized between all the resources. Now, this group, which Johan and I are a member of, is being pushed by the Human Genome, HUGO, Human Genome Organization, to try and get the standardized and accurate reporting in journals. But what we're also realizing is we need to have identical variant descriptions in the clinical databases as well. For example, you may find for a particular variant that a description is given in one format in the journal, a different format in ClinVar, a different format in LOVD, this kind of thing. And when we're teaching the clinical scientists how to identify this, they can spend hours trying to find variant information in journals and in databases, and it's all spread all over the place. Whereas if a good description had been used, a robust description had been used, all of these databases and the journals could be brought together in one place very quickly. We could identify the clinical data we need, we could speed up the decision-making process, and we can also reduce errors that are caused by inaccurate reporting. We need to get data in a shareable position. We need to make accurate data, and we need to share that data, and that will start making a big difference in your diagnostics going forward. Anyway, I think that's pretty much me done. I'm quite happy to field some questions now. I know it's a bit of information overload, but we had a lot to cram into a short session. Normally, this is kind of information taken from 2 or 3 talks and crammed together. So anything that's not clear, please do let me know.
Thanks, Pete. It's a really interesting talk. Thank you for that. We do have some questions if you'd be happy to answer them. I'm also going to unmute Nikki because I think some of these are relevant to EMQN as well. So the first question is, how should you apply this in the organisation of an EQA scheme and use in the assessment of the cases?
I think that might be a Nikki question.
I was going to say, I think that, yes, that's obviously a Nikki question. I think at the moment, Monique, I think we have to have a discussion, I think, with all of you first, because at the moment, yes, we still have guidance on LRG or on using HGVS guidance. So I think at the moment, we take our cue from HGVS, which is to use a valid reference sequence. I don't think we would go much, we won't be going any further than, we won't be diverging from what HGVS says on using a valid reference sequence at the moment. But I think I kind of felt, yes, I have been playing devil's advocate a little bit with Pete over the last few weeks. I thought it was important to be devil's advocate a little bit because I think there are some questions about how much information you can put in a clinical report and for it to be meaningful for a clinician to read. And obviously, at some point, we may need to think about that in terms of the guidance that goes into our best practice guidance, in terms of whether we need to write a little bit more in terms of the genomic, how we describe things at the genomic level, because, I think historically we've described things at the cDNA level, and the protein has been the minimum that we've asked people to do with a valid reference sequence. But that's possibly not what people are doing now. I know a lot of labs aren't that are doing NGS, they're doing genomic, but I do think that it's a discussion we need to have, but I don't think it can be up to EMQN to tell you lot. I think you lot need to tell us. And so, I'm not going to give you an answer to that today. I would say, let's have a discussion, start having these discussions in our harmonization. I think we've got our harmonization discussions coming up, and I think that'll be a good time for you lot to start giving your opinions back to us about whether we should start to move some of our guidance on these things. Is that a fair thing to say, Monique? I hope so.
I think you've answered that really well. And just thinking about time, I'm just going to try and get through a few more questions.
Can I just quickly pick up on that one as well, Rachel? I think Nick has described that very well, and I think this is something that we're bouncing around at the moment. Johan, I, and others in the HGVS nomenclature group, is that the other thing that I didn't mention in the talk is that unfortunately LRG has now been deprecated, so it is now a cancelled project. It will no longer be updated. It's going to stay static, so there'll be no more genes added. The reason for this is that they are trying to that main is taking over. So we do actually want to start, the HGVS is likely to start providing guidance saying that main should be used instead, ideally. And I think that in terms of, because this group have been really good and have said ideally use LRGs, I think that with care, main is the logical way for you to go, because it is trying to replicate and expand the ethos of LRG. And I think that if my biggest worry with this is that they decide not to create all the clinically relevant transcripts, it is an opportunity for the field to really nail this down. It is an opportunity for these big organizations to actually finally get lots of data together and tell us which are the transcripts we should be looking at when we're doing our interpretations, rather than leaving it to guesswork in different labs to do it. So hopefully that will come across, and hopefully you'll find that as you update, it's not as painful as it may sound, and you should hopefully be just as safe with main as LRG, because I think it is effectively the continuation of it.
I think this talk's great myself. I think it's really helpful. I think when I went to the main website, my kind of reaction when I first heard of main, went to the main website, was, oh my God, not another set of recommended of reference sequences. It was like, yeah. I'm confused. Why do I need another set? Because I didn't find a convincing argument on the front page, and I'm a busy person. And I know
it is helpful, and it really is that scenario where if we took main and just put the LRG badge on it, there wouldn't have been any issues. But the idea is what they're effectively doing is the exact same thing, but not building a third-party database to store the information. They're just letting you take it from source. To be honest, that's safer. Variant validator always takes its data from source. We cross-reference source information. We actually find errors and discrepancies sometimes between the internal records and things like that when we're building our data sets, and anything like that gets scrapped. So we're trying to make this easier for you guys. So anything you need, please let us know.
Right, I'll shut up now because I know there's some other questions.
Thank you. So one question that came in that it seems to have disappeared again, but it kind of follows on from what you were just talking about. Is there a main database where people can access search for main transcripts directly from source, as you're suggesting, Pete?
No, there isn't. I wouldn't recommend using the LRG website. I've seen errors in that. The ones that I know that do provide this, I mean, if you put, as I said, if you go to variant validator, just give it a couple of weeks from it to release the new build. We have got a function on it that's called gene to transcripts. You put your gene symbol in there, and it'll produce a table that tells you which is the main select. So our tool does that for it, and you can put in each gene symbol. Other ones that you can use, I know that the ClinGen allele registry provides that. ClinVar, I'm pretty sure, provides that, and that's accurate because these are NIH maintained resources. I've just seen too many errors in ensemble resources. I don't know why. They're good resources, but there just seems to be quite a few errors, and that's the thing with the LRG project. It was ensemble driven, and you just got to be slightly more careful, whereas the RefSeq are very good at their internal NIH resources do cross-reference each other pretty well. So I'd say that give it a couple of weeks for the new variant validator release and use our gene to transcript function, and that will give you a nice table, and it'll put a tick in the relevant boxes for main, select, and main plus clinical.
Perfect.
And also then if you validate a genomic description, you'll get the same options.
Thank you.
Rachel, the question did not disappear. I answered it by saying that I always go to the HGNC website, type in a gene name, and they will list the main transcript.
I forgot about them as well. Thank you, Johan.
Thank you, Johan. At the risk of starting a really long discussion when we're a bit short of time, one of the other questions that came up, which I think is really important, and maybe you have some examples, Pete, is have any diagnoses been missed, or have we unknowingly been misclassifying variants because we've used the wrong preferred transcript?
This, because we're running short of time, I kind of didn't pick up on this on my talk. And the reason that, so the BRAF gene that was the one that came up in all our discussions, the LRG has a single transcript. The main has created a main select, which would be the T1 transcript, and they've created what you would be used to calling T2, an alternative transcript. This transcript has got 2 different exons, and that's exactly the kind of question I was going to ask. A lot of labs, because of the information and guidance that's been given, would only concentrate on the primary transcript, the LRG T1 transcript. They may have never considered the impacts on this alternative transcript that's got valid, clinically relevant, biologically relevant exons in it. So the likelihood is there's a good chance we have missed diseases. That first example that I gave in the BRCA gene, if the clinician hadn't pushed hard and considered these other transcripts, even though the reporting transcript does have this particular exon, in terms of a different protein, a different transcript, that genomic variant can be more damaging. It can have more of an impact because the sequences aren't the same. Where that position, where that variant appears in the protein can be closer to the N-terminal, and there is evidence to suggest that N-terminal protein substitutions are more damaging. So without actually looking at all the clinically relevant transcripts, absolutely we can be missing things. And that's why I'm really hoping that RefSeq won't just stop at creating a main, main won't just stop at creating the select transcript, which is canonical, and another transcript just to take, just to take the additional exons. We need for each gene a full set of clinical transcripts that's based on biological evidence, loads of different evidence, people like you guys who've got interior knowledge of this about particular genes, providing that community backing and getting a really good set of clinically relevant transcripts for every gene that we really should be looking at in terms of classification of variants.
I agree.
And that's what LRG should have done. LRG should have done that. This should have been done years ago, but LRG fell short because, unfortunately, people in charge of the project decided that many LRG should only have one transcript, and then they fell short and said, no, we might have a couple for some LRGs. It was a shame. It should have been done years ago.
I agree. I can definitely think of some ophthalmic disease specific examples for where things could be missed if you don't know about alternative transcripts. So
I can think of them in terms of lots of genes, but I'm just, not necessarily LRGs, but I can think of them for lots of other genes. Yes.
So just to let everybody know, we've not got time to get through all the questions, but we are going to follow up on all the questions that have been submitted to the Q&A box. So if you think of any more questions during the session, please feel free to submit them and we'll answer them in due course. So just because time is moving on, I'm going to move on to Johan's webinar now. So we'll say bye to you, Pete. Thank you very much for your talk. Hi, Johan.
Hi.
So I'll just switch my screen that I'm sharing. If you bear with me 1 second.
Yeah, so I can explain in the meantime. I prepared some questions for everybody, because going through the HGVS nomenclature recommendations is quite boring. It's one slide with examples after the other. So what I did instead is I made a few questions which you have to answer. Answering these questions is, as was said, anonymous, and then there you can test how good you are with HGVS nomenclature. And I can also see which questions are easy or difficult and where there are misunderstandings of the recommendations. After the quiz, I have a short presentation. I will not reach the end of it, but the slides will be shared, and there will be some more detailed information on the topics where I think that you might have chosen the wrong answer here. In relation to this, I included some questions based on the questions that you have provided to EMQN, where you wanted some more guidance on HGVS topics.
Thank you. So if everybody's ready, I'm going to launch the first question. So you should now have a pop-up on your screen if you wouldn't mind answering.
Yeah, the first few questions are related to what Pete has talked, so that should be very easy for you.
The answers are coming in, so I'll just wait for it to slow down before I end the poll. Okay. I'm now sharing the results. So you should be able to see on your screens now which are the correct results.
Yeah, so a brief a G name, of course, is not cannot be used as a proper reference sequence because you don't know which transcript is used. Gene names might change. A chromosome number also not, because which genome build of the chromosome was used. And the NM provided is missing the version number. So is that version 2, 3, 4, 5? So that's also not correct. That's why none of these is the correct answer.
So moving on to the next question. So everyone should have a new pop-up on their screen. So I'm sharing the correct result now. I'm also going to try to share the overall result on screen just so everybody can see what the most preferred answer is.
Yeah, so again, none of these is the correct answer, as most people guessed. In the first case, there is no transcript identified, so you cannot use a C dot description when for the LRG you don't indicate the transcript that was used. Adding BRCA1 is not correct. That's not a transcript either. Using BRCA1 is like in the first question, it's a gene symbol, so also not specific. So that's why, again, none of these is the correct answer.
Thank you. Moving on to question 3. Which variant descriptions follow current HGVS nomenclature recommendations? And there are 4 options. You can select more than one if you wish. Okay. Oh, still some. Okay, I'm going to share the correct result now.
Yeah, to explain, the first one misses the underscore. The LRG has an underscore between LRG and the number. And answer 3 is wrong. It is not following HGVS descriptions because HGVS says you should not add something like BRCA1 in the description of the variant.
Great, thanks, Johan.
So that means answer 2 is correct.
That was a close one, that one. Whoops, I've lost my. Okay.
And I see a lot of people add the BRCA1 between brackets, but as I said, when you follow the official HGVS recommendations, you should not add the gene symbol. It's redundant information. You don't need that. And you could make a mistake there.
So launching question 4 now. This is a single choice question, so please select only one answer. Okay, looks like some more answers are coming in. Sorry. Okay, you've had a minute, so I'm going to end the poll and display the results.
Yeah, so the right answer is the second one. That's fully complete. The using one of the basic rules of HGVS is that when you use a reference sequence, it must contain the nucleotides that you describe as variant. An NM reference sequence is a cDNA type of transcript. It does not contain intron sequences. So you cannot use an NM to describe variants in the intron. So that's why the two answers starting with the NM are wrong. In addition, the third one has, again, a missing a version number. For the LRG to use, the LRG does contain intron and exon sequences, but you have to indicate the transcript that you have used. So that's missing in the first option. So only the second one is fully correct.
Okay, thanks, Johan. Question 5. This is another multiple choice answer. So please submit or select all the answers you think are correct. Oh, okay. We're coming up to a minute, so I'm going to close the poll in the next few seconds. Okay.
Yeah, so the first answer is correct. We had that in the previous question as well. The second answer not, because in this case there is a reference to the genomic reference sequence of chromosome 17, where the BRCA1 gene is located. But again, you don't indicate which transcript you use. For answers 3 and 4 are correct, because now you indicate on chromosome 17, mapping transcript NM 7298.3 or 4. You could use both. Then you can give a description using including an NM and an intronic sequence. So you do that by giving the chromosome reference sequence between in parentheses the transcript mapped on it, and then give a description using a C-dot description.
Thank you, Johan. Should we do a couple more, do you think?
Yeah, because the idea is that I cover some basic HGVS recommendations using the questions.
So I've just launched question 6. This is another multiple choice answer.
Yeah, and in the following questions, reference sequences are missing. So you can assume those are correct. So when there is not a reference sequence given, that's not a reason to call the answer is wrong.
Okay, we've got a good number of answers, so I'm just going to end that poll there and share the results.
Yeah, this question relates to what type of, say, plus, minus, star, etc. you can have in descriptions. All variants are in an intron. That's indicated by the plus or +4, +115 or -32. So that means it's in a coding DNA reference sequence in an intron. And then 6 to 4 means it's in the coding region. -6 means it's in front of the ATG. The star means it's 180 nucleotides after the stop codon, and there can of course be an intron in the 5' or 3' UTR. The only answer which is not correct is answer 3, because there will never be a C dot plus and then a number used in HGVS nomenclature.
Great, thank you. So question 7. This is another question where you can select more than one answer as correct. Okay. It looks like everybody's finished answering, so I'm going to share that one now.
Yeah, so this is based on the description of a substitution. Most people had the right answer. Everybody had the right answer. The third answer is correct. You should not use a slash or an underscore to indicate a substitution. And the first example is something that you still see a lot, that people first give the reference nucleotide, then the nucleotide position, and then the variant nucleotide. That is not correct. HGVS uses a number, a reference nucleotide, greater than sign, and then the variant nucleotide.
Okay, now question 8. Describe the DNA level variant. This is just a single choice answer, so please just select one answer that you think is correct. Okay, I'm going to share that one now.
Yeah,
Seems
so
almost unanimous.
nearly everybody had the right answer. See, so answer 2 is correct. It's a substitution from an A to a T. In theory, using HGVS, you can also describe it as an inversion of the A, or as a del ins. G dot at position 4, there is a deletion and insertion by a T. HGVS says when there are several options following HGVS recommendations to describe a variant, HGVS has something which is called prioritization. That means when this is possible, what choice do you have to make? So in this case, prioritization means a substitution. When you can describe a variant as a substitution that goes over a del ins or an inversion. So when you're interested in these rules, go to the HGVS website and look for a prioritization. Another example is when you can describe something as a duplication, you must describe it as a duplication and you should not describe it as an insertion.
Thank you, Johan. Question 9. Which is the correct descriptions of a deletion at the DNA level? This is another question where you can select more than one answer as correct. Okay. Here are the results.
Yeah, this is again based on things I see a lot in publications. The only correct answer is answer 2. Right? From 1234_1238, there is a deletion. The first one is wrong. It often comes from VCF files, I assume, where they say there is a deletion from position 1234 to position 1234. That's not used in HGVS. Then you say 1234 is deleted. Answer 3 and 4 are wrong because you should not indicate the length of the deletion, so like del 5, or the sequence that is deleted, DCCGA, because that is redundant information. And descriptions using HGVS should be as short as possible. So do not give useless information or give the same description twice. 1234 to 1238 del 5, it's obvious that there is a deletion of 5 nucleotides. But as I said, I see a lot of answers say 3 and 4. You should not do that.
Okay, question 10. Okay, that one was a bit harder to work out, I think. I'm sharing the results now, Johan.
Yeah, so this is based on the so-called 3' rule. When like in the example, there are 3 T's in a row and one is deleted, the 3' rule says you should describe the last one as deleted. So that means G.8 del is the only correct description using the HGVS recommendations. You should not use then G6 or G7, it's G8 del. Pete had an example of this as well in one of his slides, where people indicated that the first nucleotide of the stretch was deleted. The problem here is that the NGS software often uses a 5' alignment. So these tools would indicate that position 6 is deleted. But yeah, these the HGVS recommendations say you should use the 3' rule and correct that.
Okay. Next one. This is just a simple yes or no question. It's close. Okay.
This is again an example of something which goes wrong a lot. You see that also in the answer selected, because well, the correct answer is even selected a few times. When you describe an insertion, you have to be specific, and you have to indicate between which 2 nucleotides the insertion is. So the description is always, say, 835_836 ins, and then what is inserted. The problem with 865 ins G is that what do people mean? Do they mean at position 865 there is an insertion of a G nucleotide? Or do they mean after position 865 there is an insertion? So that's why the rule in HGVS is 865_866 insertion of a G to indicate where the insertion is. And from the answers you have selected, I can see that it's good that we had this question, because the majority gave the wrong answer.
Okay. Would you like to do some more, Johan? Or
Yeah, because
yeah.
I see that we cover some
some
problematic
tricky,
cases.
yeah. Okay. This is another single choice question. What is a correct description of a nucleotide tested but found unchanged?
Yeah, there were several questions on this as well in the answers you mailed me. So.
Okay. People are still answering. Maybe they're thinking about, everyone's thinking about this one a bit more.
Yeah, and I suggested after this one, we go to the presentation I prepared, but it might be good to share the other questions with the people.
Okay, that's not
after
a problem.
the meeting.
Yeah, we can do that. Okay, I'm going to share the results of that final question now.
Yeah, the correct answer is the first one. So the majority had the right answer. The problem with 1, 2, 3 A equals is, again, there is redundant information in there. And when you look at the reference sequence, yeah, you already know there is an A there, so you should not give that in the description. And the third, an A to an A, yeah, where you could say it's, you could describe a variant like that, but had the recommendation, which after a community consultation step, which I'll come back to in my presentation, was that the way that was suggested to describe such a variant is I tested the nucleotide at compared to reference sequence at G.123, and I found it as not changed. Thank you, Rachel.
Thank you, Johan. Okay.
And now I should skip to share my screen. Huh?
Yeah, if you could share your screen, that would be That would be great.
Can you see my screen?
Yes, we can
Okay.
see that. Thank you.
So I prepared a small presentation where I first will tell you how HGVS nomenclature is currently organized. So where should you go when you have questions or when you think that things need to be changed? And after that, I have, based on the questions that people asked to EMQN, some explanations on what the current HGVS recommendations are. So it all started with a paper in 2000, where the HGVS nomenclature was first published in the Journal of Human Mutation. Quickly after that, a website was started at hgvs.org. And in 2016, a second paper emerged with a summary of the recommendations. In the 16 years in between, the website was the place to go to see the most recent recommendations, and that's still the case. We cannot, when we make a simple addition to the nomenclature, publish a paper because journals are not interested in small updates with tiny changes. So an update in a publication will be rare, but when you go to the website, you always find the recommendations as they are at the moment at a specific moment in time. The HGVS Variant Nomenclature Committee is, as it is called at the moment, is a committee under HUGO, the Human Genome Organization, and that is after the last 2 years, the HGVS, the Human Genome Variation Society, and the Human Variome Project, Global Variome, have merged together with HUGO into one organization. And that's why I say now that the HGVS Variant Nomenclature Committee is part of HUGO, like the HUGO Gene Nomenclature Committee. And for instance, ISCN is also now as affiliated with HUGO as a committee. The committee is collaborating with ISCN and HGNC, the Human Gene Nomenclature Committee. And the collaboration with ISCN is, for instance, why one of the current members at the moment is Ross Hastings, who is the chair of the ISCN committee. And I am, as the chair of the HGVS Variant Nomenclature Committee, a member of the ISCN committee. The other members we have at the moment is Terence Murphy. He represents NCBI and EBI, so the big databases. Pete and Raymond Douglas are a member. Pete, you've heard earlier today, and Pete is working on software, the variant validator software, so tools to help people try and test HGVS nomenclature. Raymond Douglas is representing gene variant databases. Reese Hart from the US is representing commercial companies. And we have 3 members which are leaving that at early this year. That is Anne-François Roux. He represented the genetic testing laboratories, Peter Taschner from Leiden, who also was an LSDB expert, and Oleg Agafonov, who was an expert in standards. There was a call earlier this year for people who wanted to become a member of the HGVS Variant Nomenclature Committee, and 4 new members have been nominated, but we are waiting approval of the HUGO board, and then we will have 4 new members. And every 2 years, there will be a call for people who would like to spend their, say, spare time and help the world with sitting in this committee to further improve the HGVS nomenclature standard. So when you are interested in this topic and would be willing to serve on the committee, in 2 years we will have a next call for new members. As I said, everything is on the web page. This is just a screenshot from some of the pages. The website is varnomen.hgvs.org or www.hgvs.org/varnomen, and you will find a link to the website. I'll fly through this website quickly. One of the things is HGVS Simple under the background materials. When you are not familiar with HGVS nomenclature, it might be good to read that page The idea is that lay people should have some, after reading this page, have some clue about what HGVS variant nomenclature is, and when, because I get a lot of questions from also lay people, what information I need to be able to answer that question. So how should a completely correct and complete description of a variant using HGVS nomenclature be done? In addition, there is an educational material page where we have some slide presentations, Q&A sessions, etc. There is a glossary. On the glossary page, there is an explanation of all the terms you might find on the nomenclature web pages. So you find there the definitions of deletion, substitution, duplication, and that type of thing, and where it's explained as missense, mosaicism, mutation, etc. When you don't know where to look for specific information, you can click the looking glass. And when you do that, a page opens, you type in there a term, say deletion or mosaicism. And then when you do select the search button, it will look through the HGVS website as well as the Facebook posts we made on nomenclature, and it will give you a list of all the pages where that specific word is present, so where you might be able to find the answer to what you are looking for. We heard in the past that the recommendations change continuously. I have to stress this is not the case. Of course, after the 2000 paper was published, it turned out that we, well, first we knew at that point the recommendations were not complete. And that is because we said in that paper, we know we do not cover everything, but we suggest to start with this and then work on extending the nomenclature to cover more and more variant types and to suggest specific changes. And so after 2000, indeed, there have been some extensions to cover variant types that were not discussed in the 2000 paper yet. Of course, when there were errors or internal conflicts, we took care of those and we added on the website explanations. The only very big change compared to the 2000 paper and the 2016 paper was that at protein level, we originally suggested to use the X, and we said we should you should use the asterisk or the TAR for termination as well. I'll come back to that in a minute. So what we did do over a time towards the 2016 papers, and that was especially because people wanted to write tools on the internet to work with HGVS nomenclature. And then we found out that first we needed way stricter definitions of all the specific terms. What is meant with a substitution? What is a deletion? Very strict definitions of these cases. We had to, by that, separate the different classes of variants, so make a discrimination between insertions and duplications. That we added a hierarchy, which I also called it the prioritization. When there are several descriptions possible in theory, what is the winning description using the HGVS? And that was, as I mentioned, to make these automated error checking tools. We added versioning to support users, so to make people aware of the fact that the recommendations had changed. I'll come back to the versioning in a minute. And we simplified the use of special characters. So try to use one specific character only once, so improve overall consistency. And as I mentioned, the main change was the X, which we in 2000 suggested as to use for a stop codon. It quickly turned out that we should not use that because HGVS follows UPAC, both on DNA and protein or amino acid level. And UPAC already said the X means an amino acid changes to an unknown amino acid. And the X so stood for an unknown amino acid and not for a termination codon. Also, we found out that NCBI already in the 70s decided to describe a translation stop codon using the asterisk. So that's why we changed the X to ter for termination codon in 3-letter code, or the asterisk. So that was a big change, and well, the ugly looking guy indicates that we got a lot of bad say publicity or comments because you suggested this change. As I mentioned, we now use versioning. Top right on the screen of the HGVS nomenclature website, you will always be able to see the current version. As is mentioned here, the screenshot is older, that is from version 15.11. We currently have a version 20.05, which means it was updated last in May 2020. When there is a new rule, then we will get a new version of the HGVS nomenclature recommendations. When we add an example to the pages or correct a typo or so, that will not give a new version. It will just be a correction of the mistakes. In addition, you will be able to find on the HGVS website what has changed between version 15.11 and version 20.05. So it's explicitly shown which things have changed or have been added to the website. Before we, the committee, when we need an extension or a modification or a change of the recommendations, the HGVS Variant Nomenclature Committee makes a proposal, and that proposal will be on the web for 2 months for what we call community consultation. Anybody can then see the proposal that we have made and can send whether or not they agree or do not agree with the recommended change. So everybody, we are not adding new recommendations without asking the community to give their opinion. On the website, you will find a listing of all the proposals that we have made. The last one was proposal 010, and that is still undecided because we got some suggestions and criticism to make this change, and it is still pending. When you want to make sure that when a new community consultation proposal is made, when you go to the website, you can see that you can register at a specific email address, and that will ensure that when a new proposal is made, you get an email pointing you to this new proposal. One thing in relation to the questions I received through the EMQN is that HGVS nomenclature was designed to describe variants in DNA, RNA, and protein sequences. It's based on a stable reference sequence, and it applies to any organism. So you can also describe, when you have a reference sequence, variants in bacteria, in the COVID-19 virus, or plants, whatever. HGVS was not made for writing a diagnostic report, which more or less explains the HGVS code. Some of the questions I received go beyond describing a variant. You can often use HGVS, the HGVS format to describe what you have done in the lab, but HGVS was described specifically to describe a variant. So for instance, when there is no variant in a DNA reference sequence, in theory, you cannot use HGVS to describe that. And HGVS is not for non-experts. So when somebody has no clue what is written there, that person should also not try to draw a conclusion from this. The format, well, you have seen it. It's a reference sequence, a separator, a colon, and then the variant is described. And the variant is a mix of position or positions in the reference sequence and then the change. As I mentioned in the questions we answered, a reference sequence must contain the variant nucleotide. So that's why you cannot use an NM to describe intronic variants. One of the questions I received is how far in the intron can I use an NM to describe a variant? Well, the answer is not even one position. The NM does not contain intron sequences, so you cannot use it to describe an intronic variant. Another problem is that computer preferred descriptions of variants, look at what comes from NGS software, is a genomic description. So based on the reference sequence, a chromosomal reference sequence. So the type of descriptions you see here. The problem is that those descriptions go always into the multi-millions, and you have no clue where on the chromosome, is it in a gene, is it not in a gene, is it coding or non-coding. So that's why humans prefer the coding DNA, so a transcript-based reference sequence, where from the description you can immediately see where is it? Is it in the intron? Is it in the coding region? Is it in front of the ATG translation initiation site or after the stop codon? That is way easier. And that makes HGVS descriptions partly problematic because we have now designed a format which can use both coordinating systems. You should remember, though, that HGVS says, please describe the variant always on genomic level. And you can add a description based on a transcript level. But you can add that. It's mandatory to give the description based on genomic coordinates. And a genomic coordinates meaning a reference genome build. Well, then depending on the description, we use either a character like the greater than to indicate a description of a substitution, or in general, 3-letter abbreviations, del for deletion, dub for duplication, ins for insertion. And we also have the deletion insertion where something is replaced by something else. And but that again is in the direction of writing a diagnostic report. You can also use allele description, so indicate that variants were found on one allele only or on 2 different alleles. And as I mentioned before, also in the questions, there is some kind of prioritization. What description should be used when there are several options? One example of the HGVS variant nomenclature website, when you go to the website, choose recommendations, DNA, and then select deletion, you open up a standard page which starts with the definition. And I can read the definition of a deletion here, which is a sequence change where, compared to a reference sequence, 1 or more nucleotides are not present, deleted. And then you get how to describe a deletion. Then you get some examples of how variants can be described. And then in the discussion part, there are some relevant questions which are discussed, or we discuss, say, things that are useful to explain why we got to some specific choice. Sometimes it's a little bit debatable whether you describe it using format A or B. Then you can find in the discussion part an explanation why one type of format was selected over another type. This is the example of the definition of a substitution, a sequence change where, compared to a reference sequence, 1 nucleotide is replaced by 1 other nucleotide. So when you have a substitution of 2 T's, which change to 2 A's, then you do not describe that as a substitution. When you read the page, you will see that when there are 2 flanking nucleotides are both changed, so TT to AA, then it says you should describe that as a deletion insertion. This is then the most simple format of substitution. The substitution differs at DNA and protein level, because on protein level, we do not use the greater than sign. Probably everybody knows that. There we say the methionine at position 1A2 is changed to a leucine. On RNA level, you give the description starts with an R, and then we use lowercase letters, and we use a U for uracil instead of the T. The 3' rule, it was mentioned in one of the questions already. That's a very important rule in HGVS nomenclature. When there are 3 nucleotides in a row, one of the 3 is deleted, or there is an additional T, then the 3' rules applies, where for all descriptions, the most 3' position possible is arbitrarily assigned to have been changed. The problem recently is that with NGS sequencing and the VCF format uses a 5' aligned calling of the variant. A hot topic is exon deletions. When there is the sequence, the deletion breakpoint is sequenced, the description can be very simple. I don't know whether or not you can see my mouse, but then you have a description like G dot from position to position, there is a deletion. Or on a coding DNA level, of course, with the proper reference sequence added, 6439 minus, so from an intron position to an intron position and a deletion. However, with most of the technologies used, PCR or NGS or MLPA, the breakpoint is not sequenced, and there is an uncertainty in where the deletion exactly is. The most popular description of the deletion is then based on exon format. So you indicate, assuming that this is an example of the deletion of exon 45 of the DMD gene, where the MLPA show that exon 44 and 46 are present, then you can describe the deletion like this. The deletion starts in the intron 5' of exon 45, and it ends in the intron 3' of exon 45. The problem is, of course, that this is not what you detected. I see all kinds of strange descriptions. As I said, the top one is the exon-based description of such a deletion of one exon. I see all kinds of descriptions which are shown below, which are not correct. Of course, you cannot describe exon 45 del or IVS 44 underscore IVS 45 del. The problem with these is it's not specific. You do not give nucleotide positions, nor do you indicate what is exon 45. Where can I find how exons are numbered? So correct descriptions are shown here. HGVS has a specific page. It's mentioned at the bottom, the uncertain page, where you can find recommendations of how to describe these variants when you want to be more specific. As I said, an exon-based description of a deletion is accepted, but when you want to be more specific, for instance, when you did an MLPA, you can use a description based on the position of the probe. For the details, go to this uncertain page, and it is displayed, and then you get a description like I mentioned on the top. When you do PCR, in addition to, for instance, confirm the exon 45 deletion by doing a PCR of exon 45, then for exon 45, you can use the position of the primers that you have used. So when you use PCR, you use a position based on the amplification primers used, and you get descriptions like shown there. When you use next generation sequencing, for instance, whole exome sequencing, you can base the description based on the coverage you have. When you see that you have sufficient coverage for the positions which are shown here, it starts, of course, with 6438 plus 143, which you know is present. And then the first nucleotides which are certain, in this case 6439 minus 197, where you are certain it's deleted until the last position for which you are certain it is deleted, 6614 plus 108, and then it ends the description with the position of the first nucleotide you are certain it's present again. So for this type of descriptions, the HGVS format is give the position of which you are certain which is present, then the extent of the region which you have tested that is absent, and ending the description by the first position after the deletion, which is present again. At this point, I think, Rachel, I have to stop sharing my screen because I think we are going towards the end of the time we have today, isn't it?
Yeah, we are coming close to the end. Do you have a lot of slides left to talk about?
Well, based on the questions you have, I have added some slides, but when we share the slides, people can find the explanation on the slides. And when they have still questions on it, they can mail me, of course, and ask me to answer those questions in particular. But maybe it's more fruitful when we see whether or not there are any general questions on HGVS nomenclature.
Okay, if you're happy to do that, we can do that. So
Yes.
I'll just reshare my screen. So if anybody's got any questions for Johan in this moment, if you wouldn't mind either raising your hand or submitting them to the Q&A box. We have got a few questions that have come in during the talk, Johan. The first one is it possible that you give a subversion to the HGVS page if you do corrections? Because people would like to see a list of corrections made with each version release.
Yeah, so HGVS recommendations are also are on GitHub. I assume not everybody knows GitHub, but when you go to GitHub, it's a source which say computer programmers use to show all the different versions of their software. On GitHub, you can find a copy of every change that was made to a page on the HGVS website. And so when you want to see when we corrected the typo or whatever, you can find every change, even when I changed a period into a comma, you can find that back using the GitHub resource. When people can't find it, please mail me and I'll give you the URL to this resource. So yes, you can find every change back. For changes in version number, they are on the website listed specifically. But when we added an example or corrected the typo, to see that, you should go to the GitHub resource and you will be able to find all the changes in the pages there.
Great, thank you. I think people will find that really useful. The next question is, how would you describe a deletion if it's the first or the last exon that is deleted?
Well, you can do that in the same way as I indicated. I can imagine that you tested exon 1, which is negative, and you do not have a probe which is 5', which is tested positive. In that case, the position of where the deletion started is simply a question mark. And so the description starts with something like C dot parenthesis, question mark, underscore, and then the position of the probe that you have tested, which is deleted. But there are examples on the HGVS nomenclature deletion page. There are examples on a deletion of an internal exon, the first exon, the last exon, any option you can imagine. You can find an example on the HGVS nomenclature website.
Okay.
Both, by the way, for deletion and duplication.
Thank you. Thanks for that. This next question is about how to describe protein changes. So if there is a frame shift at the DNA level, how would you describe the protein change, i.e. should you give the detailed description of the protein change, something like P dot brackets GLU 1234 allo frame shift star 12 close brackets, or should you use P dot 0 question mark?
Yeah, I can see, by the way, the question on my screen also. So, but HGVS recommendations say you must describe a variant based on DNA level, and you can add predictions of what you observed on RNA and protein level. So the easy answer is it's not mandatory following HGVS to give a description on protein level. When you give a description on protein level, it is what you expect or predict that will happen. And whether or not you use then the extensive description, which is the GLU 1234 ALA FS star there, for when you think it's a frame shift, when it's a prediction, because you have no RNA or protein data, you should give it in parentheses. And then HGVS has two formats, the short form, where you simply say glue 1, 2, 3, 4 FS, or it has the long form, which is in the question. But it's up to you to use either of the two. HGVS thus allows both. The only thing which HGVS says is you must give a description on DNA level, and you are not allowed to give a description on protein level only.
Nikki, I just brought you in because I wanted to say, because the question is actually based in the context of marking EQA schemes. So I was just wondering if you wanted to input anything to answer that question.
Yeah, well, I must admit, I haven't looked at the specific example in great detail, but I can see Stacey came up with a sort of comeback second question on it. And I think she's stating that the labs are saying that the protein undergoes NMD. So perhaps I think in that case that you'd certainly give a comment that you would expect the nomenclature to actually, you know, be consistent with what you're saying the protein's doing. So I don't think we would probably, you know, that we would give some advice that if you're saying that the protein's going to undergo NMD,
yeah, so when you
saying that it's a termination is probably that predicting a termination and then saying it's undergoing NMD is not logically consistent, is it? So probably a comment on that would.
Yeah, when your prediction is that the protein and well, the RNA goes NMD, then you can give the description P dot between brackets O question mark,
Yeah.
where you indicate I assume that the protein is not generated. That's because it's a prediction. Following HGVS, you can give such a description. Yeah. Whether or not EMQN accepts that as correct is not, that's not something that HGVS should decide.
And I think because it's prediction, we are rather, you know, we tend to do this, as I say, we tend to be quite not lenient, but we take this, you know, we take it seriously. But I think it is a prediction. But I think it is logically not consistent there. So I would certainly give the lab some advice to be a bit more logically consistent. That if you're saying that the protein's doing one thing, that it would be sensible to have your HGVS saying that it's doing the same as what you're saying in your report.
Okay, I think you've both answered that question quite well from a HGVS perspective and from an EMQM perspective. So I think because we're already over time, we might just do one more question.
Yeah, the first question I see is there was no dot on the slide. That is then a typo from my side. So okay. The question from Martina.
Okay. If the whole gene is deleted, would it be possible to use some kind of nomenclature indicating that the deletion could include flanking genes?
Yeah, in the slides that I prepared, people will find, probably it's the next slide or the one after that, where I have had some examples on this. You always indicate the first probe present, then the second position is the first probe you detected as negative, up to the last one, which is negative, and then you give the position of the first probe, which is positive again. That's the standard description for any type of deletion using HGVS. And whether or not it's an exon or 2 exons or 1 gene, 2 genes, 10 genes, the format is always the same.
Okay, thanks very much, Johan. Thanks for your really interesting and informative talk. So I think because of the time, Nikki, if you wouldn't mind closing the session, would that be okay?
Yeah, that'd be absolutely fine. I'm still unmuted, so that's fine. Brilliant. So yeah, I'd just like to thank both Pete and Johan for giving up their time today for these webinars. I think they've been really interesting, and I know they're going to be the further topic of discussions at our harmonisation sessions that we're going to be having in our assessment meetings. So I'd like to thank you both for giving such clear and illuminating presentations. I think we've got a lot to think about, and I'd also like to thank our audience for joining in the webinar and submitting some really interesting questions. I know we're going to have to come back to you on some of them, but yeah, we will come back to you. This is a new format for us, and I think it's really exciting. And you know, I think we're hoping we're learning from this experience, and we're going to try and put on some regular webinars on topics of interest to our members, such as sort of things like the new best practice guidelines, new technologies, educational topics, etc. And finally, I'd just like to give a thank you and a mention to all the EMQN staff that have worked so hard behind the scenes to put this webinar together. Something like this, a venture like this, it doesn't come off without any planning and practice. So I'd like to give a special mention to Rachel, who hosted today and coordinated the program with Johan and Pete, and Katie, who's been our sort of technical and project lead, and all the Germline team who've helped run the back room of the meeting. So they've been sorting out the questions and things and putting them forward. And they proposed having a webinar to replace our old-fashioned guest speaker at the assessment meeting. So I'm going to stop there because you've probably heard enough from me. I'm always yakking on. But I'd like to thank you all again for coming to this meeting. And I really look forward to seeing everyone at the Germline Assessors meeting in the next week or two. And you know, that's virtual again for everyone. But for any Molepath assessors who happen to have come along today, we're still looking forward to seeing you face to face, maybe at the end of the year. We're keeping our fingers crossed. But thank you all for coming. I'm really looking forward to everyone, and thank you very much. I've had a brilliant time.
So I'd just like to say before everybody leaves that we are collating some survey questions just as you exit the webinar, and it's really useful for us to have that feedback from you so that we can make these sessions more useful for you in the future. So thanks in advance for completing those questions for us. And with that, I think we'll all say goodbye. Thank you very much, everybody.
Bye.
Bye.